In this video, we will be learning about microscopy exam questions. By the end of the session, you will be able to define different command words, you will be able to read A-level exam questions, and confidently answer calculation questions. First of all, before you do any A-level exam paper, you should know what your exam words are asking you. AQA uses loads of different command words for their questions, and the full list can be found below. Make sure you write down that web address, so pause the video and then you can find that full list so that you know what command words you may be faced with. However, I have picked a few that I think come up a lot for you to look at. On the next slide, have a look at the command words and check you understand what they mean. So write down the definition for each command word, and then you can check your answers on the next slide. So here is what AQA mean by their different command words. So to apply means to put into effect in a recognised way. So apply your knowledge to a particular question example, okay? Calculate is to work out the value of something. To compare is to identify similarities and differences. You will not get the answer or all your marks if you only identify similarities or differences. Define is to specify the meaning, so give a definition of. Describe is to give a really basic account of, so just tell me what's happening. Evaluate is you might get some evidence, so that could be data, and you'll be expected to judge from that data. Explain is to give reasons, tell me why. Identify is to name or otherwise characterise. Predict is to give a plausible outcome using your knowledge. And suggest is to present a possible case. However, you must draw on your scientific knowledge for this. Now, if you would like to know more about application questions, there is another PowerPoint which is about application questions. So do watch that to help you answer those. But today, we will be focusing on calculation questions, so working out the value of something. So microscopy exam questions are often hidden inside other questions. The first question we will look at is going to be a microscopy question that is hidden inside another question about red blood cells. You can answer them confidently once you've learned to break them down. So we're going to go through them together now, and let's have a go at these microscopy exam questions. So here we go, we've got a diagram of a red blood cell and an alveolus. It doesn't tell you it's a red blood cell. So it shows you an electron micrograph of a section through parts of an alveolus of a lung. X is the inside of your alveolus. And it asks you A, to describe the path of a molecule of oxygen from the air in the alveolus of X to the plasma membrane of A. So you need to describe how the oxygen gets from X to A. Pause the video and have a go. So you need to use really detailed biological words. So the first place that oxygen molecule is going to go through is the epithelium, which is underlined. You would not get the mark if you did not put that word, of the alveolus. It will then go through the capillary wall, or the epithelium, or its endothelium, and it's going to go through the blood plasma to that cell. You need all that for 1 mark. Now, if you look at the examiner's report, you can see where students commonly fall down, and it's really helpful to do this when you're revising so that you do not end up in the same position. So only the best candidates interpreted the diagram correctly. So if you found this hard, don't worry, a lot of other students did. You must use appropriate biological language, that's epithelium, endothelium, okay? They weren't happy with references to walls and membrane because they weren't sure of your understanding. In addition, students would talk about the mechanism involved. Now, they won't just describe the pathway, which is what it's asked you to do, you've tried to explain the process of diffusion, but that's not what the examiner was asking you for, so you wouldn't have got any marks. So B, cell A is a eukaryotic cell. Give 2 features that may be found in a prokaryotic cell, which are not found in cell A. So pause the video and have a go. So it lists here some things that are found in prokaryotic cells but will not be found in cell A. So that's your cell wall, capsule, flagellum, mesosomes, plasmids, genetic material not inside a nucleus, so it could be a nucleoid or free DNA, and ribosomes. Now, most people got having as a capsule and a cell wall correct. However, they said there was a disappointing number of incorrect responses. And this was because the candidates sought to compare a prokaryotic cell with an unspecified eukaryotic cell, or they made references to eukaryotic features such as nuclei and mitochondria. These are absent from prokaryotic cells. This was probably because candidates didn't read the question properly. So that's an important message. It's asking you what's not in a eukaryotic cell, what's in a prokaryotic cell. Don't get it wrong, read the question. So the next one was cell A and B are biconcave discs, and it asked you to explain one example of a biconcave disc over a spherical cell of the same volume in transporting oxygen. So pause the video and have a go. So it's because a biconcave disc has a much larger surface area than a sphere. Now, to get the second mark, you must have linked this to diffusion or said there's a short diffusion distance. So large surface area, always link that to diffusion. Don't put one on its own, because otherwise you won't get that second mark. So it said candidates actually did this really well. So if you could explain surface area, you can link that to diffusion. So make sure you are able to do that. However, others were unable to explain the flattened shape. Always think in terms of surface area when you were talking about diffusion and diffusion pathways. And they might have constructed inappropriate arguments based on the amount of hemoglobin. So do not make assumptions, use the knowledge that you have and use that to answer the question. So finally, we get to our magnification question. So you can see how it's hidden inside your exam question on red blood cells here. So it says the diameter of a human red blood cell is 7 micrometers. So calculate the magnification of a drawing and show your working. If you were doing this for real, you would measure the diameter of your red blood cell. But I have done it for you. So I have measured the diameter, which is the widest part across of your red blood cell. And that's 55 millimeters. So work it out from there. Pause the video and have a go at working out what the magnification of that drawing is. You should have got an answer of 7,800 to 8,000. Okay, now this should have been achieved by doing 55 times 1,000, because you need to work it out in micrometers to get the magnification, and then do 55,000 divided by 7, which gets you just over 7,800. If you divide it by 7, you would have got a mark. So if you don't know what to do, if you measured it incorrectly, you would have got a mark. But you should have done 55 times 1,000, so you get 50,000 divided by 7. The second one is in calculating the magnification. What assumption did you have to make about the section that was cut? So pause the video and have a go at answering that question. It was the idea that it's been cut through the maximum diameter or the middle of the red blood cell. So it wasn't cut a third the way along where it'd be shorter, it was cut through the maximum distance. So 55 millimeters was the maximum diameter of that red blood cell. So in your part 1, candidates only had limited success. So you had to select the right measurement and then you had to convert it to micrometers. Now both tasks presented a limited ability, so make sure you do plenty of practice on this. So make sure you are good at measuring, identifying where you need to measure and conversion to micrometers. Further difficulties then arose from a failure to calculate magnification, so make sure you know how to calculate magnification. Part 2 was answered well, okay, but there were those who didn't think about the orientation of a specimen. So make sure you think about orientation, where a mitochondria can look round because it's been cut through lengthways rather than oblong, where it's cut through horizontally. So always think about that. Okay, let's move on to another question. So this one focuses more on the process of microscopy and also makes you use a scale bar to work out magnification. So, a scientist examined the structure of mustard plant leaves. He viewed temporary mounts of leaf tissues with an optical microscope, and the figure below shows a drawing of typical results. It wants you to describe how temporary mounts are made. So think of your onion cell practical. So this is where practicals will come into play. They don't always ask you directly, but they do expect you to know what temporary mounts are. Temporary mounts are microscope slides that you only use temporarily. So for example, the onion slide, you'd make that and then you'd wash it and use it again. So to do this, Your first mark is for taking a thin slice or section. The second is for putting on a slide of water or solution or a stain, so you might have said iodine stain. And then the third one is to add your cover slip. Or some schools will put it between two slides, but generally you add a cover slip. So you'd say it's thin, and you say you add water because the water helps it hold to the slide, and it also helps your cover slip to stick, and you need to mention that cover slip. You have a maximum of 2 marks. So you said it was thin and you added a cover slip, then that would have been okay. So let's see how students did with this question. So few students apparently had an understanding of the term temporary mount. Some students thought it was a structure inside the leaf. So it suggests that practical work isn't a common activity. However, it could just be the students forgot. So make sure you know your practicals inside out. So make sure you engage with them in class, and also make sure that you revise your practicals. Watch YouTube videos on your practical. Often, vague references enable some to accumulate marks. So if you said rectangular glass pieces and cover slides, they think they gave you the benefit of the doubt. That won't always happen though, so do know the proper words of things. So B, this time it's asked you to calculate a distance in micrometers between G and H on a leaf. So I've put the measurements for you. So between G and H is 64 millimeters, and the scale bar is 16 millimeters long. So work out how far G and H are on the leaf. So the actual distance between G and H. Pause the video and have a go. So you need to divide the image length by the key length. So you know it's 64 millimeters divided by 16. So it gives you a factor of 4. So G to H is 4 times bigger than your scale bar. You know that your scale bar represents 50 micrometers. So you need to times that by 4 to work out the distance between G and H, which should give you 200. They're generally very kind, and they've given you a range of 185 to 217 micrometers. Most students did well at this, so make sure you are good at these types of questions. Next question is to describe how the scientists could have used the temporary mounts of leaves to determine the mean number of chloroplasts in mesocell cells of a leaf. Pause the video and have a go. So you need to select a large number of cells, and you do it randomly. So you're taking a sample, you count your number of chloroplasts, and then you divide those chloroplasts by the number of cells in your sample, because that gives you a mean of the amount of chloroplasts per cell. Excuse me. Surprisingly, only 5% of students achieved full marks on this question. There was confusion about the meaning of temporary mounts again, and also some students would estimate the total number of chloroplasts in leaf by counting all the cells and extrapolating the number of chloroplasts in one cell. Hardly any students said to pick your cells at random, so always think about taking a sample when you were trying to find a mean. They also didn't say, because they weren't thinking about taking a sample, they didn't say choose a large number of cells. And it was achieved for making a statement about counting chloroplasts was the only mark that was generally achieved. So you should now be able to define different command words, read A-level exam questions, and confidently answer calculation questions.